Welcome to a lecture for my Bio to 40 students. Today we're going to be talking about Unit 27-28. We have already done Unit 27 lecture. So as I said before, we're going to need to finish up this chapter. That's what we're going to do today. And then start a new chapter on embryogenesis and fetal development. I'm basically talking about pregnancy. Now, the video recording for these will be available online, so you can watch them as necessary, and all the copy of the notes will also be provided to you. Now, for this chapter, essentially, what we want to go over is gametogenesis. This is the process of forming the gametes. The gametes are sperm cells and egg cells made by the male and the female. So we have already reviewed the anatomy of the male and female reproductive system. So now we want to continue just talking about essentially where are these cells forming, what happens, where the naming of the cells, and then explain how this happens in terms of the processing for the actual fetal development after fertilization. So keep in mind, essentially, for this process, this is the special type of cell division called meiosis that's taking place. Meiosis is different from mitosis, which is regular cell division. This is cell division, meiosis, only taking place in the gonads, meaning the testes and the ovaries for the female, and ovaries in the female, testes in the male. And these are when cells are, instead of being split into 2 different cells, they're actually split into 4 different cells because we do division twice. So this process, make sure to look up in the book what it looks like in picture form, but essentially it's very similar to regular cell division, except division is taking place twice. Certain other differences I'm going to outline here as well. Now, let's go over the male part first. Right, so for the testes, we have a process called spermatogenesis, or formation of the sperm. Essentially, what that means is that these are organs that, using different hormones like testosterone and others, direct the production of the sperm cells, which are essential for reproduction. Now, where do we get testosterone? Testosterone is actually produced by the Leydig cells in the testes. So they're specialized endocrine type cells in the testes producing testosterone, right? The testosterone is needed for all the normal sexual characteristics in the male, for the production and functioning of the reproductive system, obviously, and normal formation of the sperm. So when the sperm forms, you'll see in the book, they'll show you pictures of multiple different steps involved. I'm going to summarize this here to keep in mind that at first we start with what's called diploid cells, and we wind up with the haploid cells. Diploid cells are the cells that have the full number of DNA. Haploid is the half number of DNA. Remember, we need to have half number of DNA for fertilization to occur, because 2 cells, when they meet at fertilization, they need to combine to give us full number of chromosomes that we have, normal DNA for that organism. Okay, so in our case, we have 46 chromosomes, or 23 pairs. That means the sperm has to have 23, and the female egg cells need to have 23. When they combine, you get 46 to get back that normal number for the human cell. So spermatogenesis continues in this form. Again, it starts with formation of spermatogonia, which are diploid cells, then goes to becoming spermatocytes, then spermatids, and actually finally sperm cells. Once you reach to this level, these are now haploid cells, so 4 new cells produced. And these cells, again, are essentially there for fertilization, right? They will be coming out from the body during sexual reproduction to meet the egg in the female reproductive tract, and to join with the female egg to produce a zygote, which we're going to talk about soon. So 4 cells are produced, very important to keep this in mind. Okay, so for oogenesis, for the same process taking place in the ovaries in the female. We have granulosa cells there. They secrete estrogens. Estrogens are the similar hormone to testosterone, except here they direct all the secondary sexual characteristics in the female. This is development of breast tissue, development of all reproductive organs in the female. And so estrogens are very, very important. And again, they're produced by the ovaries, by the granulosa cells. Now, normally when you look at the ovary, you see that they contain multiple ovarian follicles, and these follicles are at different states of maturation. And what's very important to keep in mind here is that unlike testes, which are producing sperm continuously, and essentially as long as the male has reached puberty, the sperm will be produced by the testes for the rest of that person's life. In the female, the process of oogenesis actually takes place in a very different manner, and basically starts actually before the female is born, okay, and then actually only finishes completely when that egg has actually been fertilized by the sperm. If the egg has ovulated, came out from the follicles, but has not been fertilized, it actually doesn't complete all of the stages of oogenesis. All right, so basically the process, again, very similar to formation of sperm, except here we call it oogonia instead of spermatogonia, and we have oocytes and then ova, the actual female egg cells. When they say female egg cells and ova, right, or ovum, that is the same thing. So it actually should be ovum here, technically, since it's only one cell, right? Ova is technically multiple. The rest is what's called polar bodies, right? So only one cell is produced is important, and that's because we want to have only one good cell with all the resources, all the cytoplasm, all the resources in order to continue with the pregnancy after fertilization. The other cells are discarded, right? They will have the DNA, but they're going to be removed as what's called polar bodies. So again, for the male, we start with one cell, produce 4 sperm cells. For the female, we start with one cell, but produce also only one cell because the rest are discarded as polar bodies. Now, what's interesting here, like I said, is that the meiosis 1 stage, right, this first stage of meiosis, actually begins during fetal development and then is then sort of paused or arrested and then restarted at puberty. And then again, when metaphase 2 is basically halted until fertilization occurs, right? The best way of thinking about this is that we only want the egg cells to undergo the entire process, only those that will actually meet the sperm. Those cells that have came out during ovulation and will not meet the sperm, they can be washed out of the body and will never actually complete this process. So just in summary, what happens in meiosis, basically we have meiosis 1 and meiosis 2. This is essentially thinking of mitosis taking place twice. Prophase, metaphase, anaphase, telophase, right? Remember those stages like we learned in the previous semester. Same thing here. The only major differences is that, again, the division occurs twice instead of once, like in regular cell division, and that we also need to have some mixing of the DNA, right, to favor the formation of successful and healthy offspring, right? So there's something called synapses taking place. That's when the chromosomes kind of line up tightly, closely together, and crossover when they exchange some of their genetic information. And so each of those new 4 cells forming, like for instance in the sperm, will actually have some mix of DNA, so they're not identical like from the parent, like there wasn't cell division. That's mitosis. Remember, mitosis, split cells, let's say for instance skin cells that are formed, are exactly pretty much the same as the original parent cell, while in meiosis, the offspring, the new cells produced are slightly different. Right? And obviously we're always forming haploid cells. Now, for the next process for this chapter, for embryogenesis or formation of the embryo and fetal development, this is essentially an explanation of how pregnancy takes place and what happens at the point of fertilization and later on in the woman's body. Obviously, primarily this is taking place in the uterus, so refer back to the video and explanations taking place for the previous chapter on the reproductive system anatomy, the male and female reproductive anatomy. Now, so very importantly for the formation of the successful formation of the embryo and then the fetus and the child is the formation of the placenta. The placenta is a very interesting organ, is that it forms actually from combination of tissues from the mother and from the developing child. Right? We call this type of unusual cell the trophoblast. Essentially, the way to think about this as the collection of cells after fertilization traveling through the endometrial cavity in the woman, actually, and as it's trying to implant and start to develop, actually is forming these new cells and allowing them to nutritionally and physically support the developing embryo. So placenta will be made out of what's called extraembryonic membranes. Two primary membranes exist. One is the chorion and the other is amnion. Chorion is the sort of outer layer, amnion is the more internal layer. The chorion is known for its chorionic villi, which are like finger-like projections of blood vessels that aggressively kind of dig into the endometrial cavity of the uterus in the female to actually provide the nutrients, right, and eventually form that deep connection between the mother and the fetus, right, through the umbilical cord, and making sure the mother is providing the nutrients to the fetus. And the amnion eventually produces the amniotic fluid, which is that liquid that we know bathes the baby inside the mother, especially noticeable later on in pregnancy, during the later stages of pregnancy, when the baby is essentially swimming inside amniotic fluid, and offers protection and cushioning from impact to the baby, right? Sort of like the brain is cushioned by the cerebrospinal fluid inside the skull, so that during any kind of impact, it's not actually hitting against solid wall. Similar, right, we're offering protection like that with the amniotic fluid to the child. So I'm going to be using, by the way, the word embryo and fetus and kind of baby probably sort of interchangeably, although there is separate sort of definitions for them. But for the purpose of this discussion, I'm going to kind of interchange these words. Now, obviously, again, the umbilical cord is essential. That is the ultimate connection that we all have from the mother to the baby, and all mammals have umbilical cords, right? These are blood vessels, arteries, and veins that provide all the nutrients and oxygen to the developing fetus before they're ready to breathe on their own outside of the mother, right, after birth. Now, in order to understand embryology, what happens, right, basically the study of the embryo, we're going to look at germ layers, the gestation, and the fetal cardiovascular shunts. So germ layers are thinking about when the fetus is getting developed, right? We start essentially one cell, but somehow eventually wind up with millions and millions and billions even of cells later on and in life. And so how do we go from that, right? The way to make sense of it is that we have realized after many decades of research is that there's actually these separate lines of differentiated cells that are created during embryo formation, and they're separated in these three germ layers. And the cells, basically governed by the genetic code, are essentially told where to go in the body and what organs to eventually become, what tissues, what histologic layers to turn into, and eventually, right, give rise to all the organs. So for us, when I'm naming these germ layers, you need to know the names of the germ layers and just a little bit some examples of what kind of eventual organs they will become once all the organs are formed, once organogenesis is complete, which is formation of the organs. So we have three layers. Basically, they start as just very simply, you're thinking sort of like a ball of cells. The outer cells are the ectoderm cells, the deeper ones are the mesoderm cells, and the deepest layer is the endoderm, right? So three layers: ectoderm, mesoderm, endoderm. Ectoderm against the outer, so will eventually give rise to epidermis, or the top layer of the skin, neurons, right, spinal cord, the brain, basically all the parts of the nervous system, and then melanocytes, which are the melanin or pigment producing cells in the skin, also actually coming from the ectoderm. For the mesoderm, we have the middle layer, so muscles, like skeletal muscles primarily will be formed here, bones, right, so all of the skeletal system, and even the cardiac muscle, all of the muscle basically inside, right, part of the heart is coming from the mesoderm eventually. And for the endoderm, the deeper layer, the best way of thinking about this is that the digestive system organs or gastrointestinal tract will be formed from there. So the stomach, liver, intestines, definitely coming from the endoderm, and that's what we want to remember here. There are other examples, you'll see them in the book. Know the ones that I'm talking about here. Right? As always, we're focusing on the actual discussion that I'm providing you here. Right? You are welcome to look at more information in the book and study that as well. Now, for the gestation process itself, right, the gestations or the pregnancy, we don't say the woman is pregnant for 9 months. We say she's pregnant from 37 to 42 weeks primarily. So as long as the baby is born within that period, right, that's term. Anything before 37 weeks is preterm. So normal gestation is around this time period, right? So most women give birth around 40 weeks of gestation or so, although obviously there's all kinds of different differences, but there's it's kind of on the spectrum, so there's definitely quite a lot of women that give birth preterm, and other times right now we have the medical technology to deal with those babies to help them survive better, things like that. And it's actually impossible to be born post-term as well, post these dates. So for the normal development, essentially we are talking about here formation of the zygote in the very beginning when the egg and sperm have met. So when they meet, right, this usually happens inside the fallopian tube, and as all the sperm are rushed towards the egg, when one sperm enters, the tail falls off the flagella, only the DNA enters and migrates to the DNA of the female and then joins together, and then immediately sort of this reaction occurs in the cell membrane of the egg so that no other sperm can join in. Obviously, we don't want that happening so that no additional DNA is provided, not to hurt that cell. And then so that new cell that has been fertilized is called the zygote, and it starts replicating by regular cell division, by mitosis, until eventually it gets to a ball of cells called the morula, and that's around 16 cells in size, and then continues to progress in more. Once it gets a little bit even bigger into what's called the blastocyst, this is a ball of cells that actually has like a cavity inside. This is the one that on about day 6 after fertilization gets deeper into the endometrial cavity and implants inside the body of the uterus in the female. Remember, we do not want implantation to take place anywhere else but the body. We don't want it close to the cervix, we don't want it in the fallopian tubes. Any kind of implantation that might occur and development of the embryo outside of the proper endometrial cavity will be called ectopic pregnancy. That is really bad for the mother. Basically, that means the baby cannot survive, and it's going to be essentially like a miscarriage. So ectopic pregnancy again means if the embryo starts to implant somewhere else outside of the proper endometrial cavity. So for normal development, right, continues on this way. So early stages, we call this the embryo, and then once more organs start to forming, we eventually becomes a fetus, and then upon birth, right, the newborn is called a neonate, and at that moment when the baby is being born, during labor process, right, so the baby is expelled through the vaginal cavity, right, kind of pushed, squeezed through, and takes its first breath. This is where we want to keep in mind about these fetal cardiovascular shunts. So the cardiovascular system, even though it's essentially the same as in the adult or in the normally functioning person, right, during the fetal development, when the baby is relying only on the nutrients from the umbilical cord, it actually is not really using the lungs, is not using the heart the same way. So these shunts exist, or basically these unusual connections in the blood vessels. We're going to talk about 2 of them. They allow for the blood to be going from the mother to the baby, essentially in such a way so that the baby gets all the nutrients, and because of the kind of unusual situation of being connected to the umbilical cord inside the mother. So two important connections is that there is a connection called ductus arteriosus. That's the first shunt. It's basically a connection between the aorta and the pulmonary artery. Remember, pulmonary artery normally just goes from the right side of the heart to the lungs to get the oxygenated blood there. But because lungs do not really function in the developing fetus, we don't need to send the blood there. So instead, they're connected, and the aorta basically is connected to pulmonary artery. We call this connection ductus arteriosus. Once the baby takes its first breath, connection closes and eventually becomes ligamentum arteriosus, which is like a strip of tissue, and then the normal anatomy is reestablished for all the organs to function because then the lungs inflate upon the first breath and can start getting the oxygen, and the blood flow needs to be going through the lungs, obviously, to oxygenate the blood and allow the cardiovascular system, pulmonary system to function normally. And then another connection that's unusual is there's an opening in the what's called the intra-atrial septum, or basically the right and left atria in the heart. Normally there is a wall, the blood isn't mixing, but in the fetus it is mixing. We call this foramen ovale, this opening there. And this opening again will close when the baby takes its first breath in the first few hours and days in life. There are some people who continue having this opening if it doesn't close properly. If it closes, it's called fossa ovalis. If it doesn't close, it's called atrial septal defect, ASD. And essentially, some people who have that, have some mixing of the blood, have symptoms of anemia, as they're sort of providing less oxygen that they're carrying because of this mixing of deoxygenated and oxygenated blood. Again, very important for this, make sure to look in the book, in our textbook, in the lab manual, to see all of these pictures and follow along with the notes.